pull up a chair, take out the oxygen, and join the discussion.
You've just stepped into life unculturable.
The world where conversations grow faster than colonies.
We're your hosts, Sam and Julia, scientists by day,
Podcasters by night,
and professional oxygen avoiders 24/7. Join us in exploring the science of microbes that thrive in the absence of oxygen on Life Unculturable.
Well, hello and welcome everybody to the next installment of Life Unculturable. We're back in the podcast chamber, Sam and me. And today, what are we going to talk about today, Sam?
Well, Julia, first and foremost, happy to be here. Nobody's canceled us yet, so that's good. We're just clicking right along. And today, I'm super stoked because we're going to be talking about a couple of really important principles of what we do in the lab. And that is the enrichment and the isolation efforts with respect to our obligate anaerobes.
Mm-hmm. I like the way how you always said it's our bread and butter. It's like the thing we do most in the lab and what our lab is most known for too, right?
Yeah, I would think so. Yeah, even I think before coming to OSU, both Mustafa and Noah were still probably regarded in that capacity.
Yeah.
So.
Especially in the anaerobic fungi world, but also we've talked with Noah and Mustafa recently more in depth about their bacteria stuff that I've completely ignored because I don't care about bacteria.
Oh, if only I could ignore the AGF.
Oh yeah, no.
If only.
We're everywhere and we won't shut up.
No, well, that's true. More about the diva-like AGF later on, folks.
So in our podcast, we've talked about what the lab in general does and what sample procurement is for us and how you are the OG of sample collection. And today we're going to talk about what we're doing with the samples once they're in the lab, specifically microbiology-wise, like not molecular work, but microbiology work.
Ooh, so we're keeping it mostly culture dependent then.
Yes.
Yeah. Oh, that's fascinating. And that's a good point, Julia, too. Like, it's great to get a lot of samples in, but it doesn't really mean anything if we don't do anything with the samples. And so, yeah, once we get them into the lab and kind of get them in the queue and into the chamber and into the tubes, that's really where the magic can potentially start to happen.
Yes, absolutely. And I mean, of course, sometimes culture-independent work comes first because our lab does a lot of this too. With the samples coming into the lab, we send them through our amplicon sequencing pipeline to even find out what's in there. But the main focus is getting them to enrichments and isolation and trying to get novel organisms. In your case, bacteria. In my case, fungi.
Definitely so. And I like how you put that. I don't think that we can really, in the case of our lab, talk with one without the other, whether it's culture independent versus culture dependent, because there certainly are parts to both. Mm-hmm. And especially for the AGF with the amplicon sequencing, that's a gigantic part of what you all do and how you sort of pick which fecal samples you want to work with in the first place.
Yes.
Yeah. So Julia, that brings me to the first question that I sort of wanted to ask you about, and that is essentially like when we're enriching, right? We're trying to hone in on a specific microbe. Like you may have done some culture-dependent survey of, say, your fecal samples, and then you get the data back, and you notice, oh, out of the 20 samples that we sent off, really only 5 of them are worth working with. Sort of why would you justify those 5 as being worth working with based on your sequencing data? Like, what are you looking for in that sequencing data?
So,
what's that tell you?
In our lab, we mostly hunt down novel strains for the most part. Sometimes we have like a research question that requires us to get some already known strains. But in either case, we try with culture-independent work to identify which AGF, like which strains are in a specific sample. And usually each sample only has a handful of strains. Sometimes some samples also only have like one main strain that you find in there. And then we're going to pick the samples that have the one strain we want or that gave us the most uncultured strains, like unknown, unclassified strains. And then we go to that sample to hopefully pull out this novel organism and then isolate it and describe it.
Wow. And so you're talking about sort of like the relative abundance of
Yes.
that particular amplicon in your sample.
Yeah. I mean,
Yeah.
it's like, it's not a very accurate science of like, because you have a 50 ml tube of poop sometimes with like horses and cows. It's easy to fill like a whole 50 ml of those. And then you take, 100 milligrams for amplicon sequencing.
Yeah.
But we found, or in my experience, the 100 milligrams that you take out of this poop is usually still quite reflective of what is in the sample. I've yet to isolate from a sample that we did culture independent work on previously, where we've not identified the strain that I'm going to pull out.
I was going to ask you that question, like if you ever actually enrich for things that you didn't necessarily see in your amplicon data.
Yeah.
Because that's something that will happen frequently with respect to the bacteria, especially if we're just talking like amplicon sequencing. And I was wondering how that would compare to the AGF. I was suspecting that it would be a little bit less ambiguous for you AGFers. So.
I think the main issue or the main reason why we do it, like why that happens for us, is not that the sequencing is so accurate that we only identify what's really in the sample. It is more so that when you enrich, you usually only pull up like the top 5 things you identify, so the ones that have a relative abundance of above 50%. Sometimes, I mean, you can sometimes pull out stuff that is lower in abundance, but usually you only get the very abundant ones. And those abundant ones, to not identify those really abundant ones in the amplicon sequencing, something has to go very wrong
Okay.
in your sequencing run.
Okay.
Although, I mean, it's not 100% accurate. So I'm sure there are people out there where they've pulled out something that they have not previously seen in the sample if they're doing the same approach.
Interesting. So we've looked at now our data that's come back to us. We've thrown out 15 of our 20 tubes. We've got the 5 that we want to work with. Now we have to start thinking about making enrichments. And so for those of you that may not be familiar with this idea or this concept, essentially enrichments are sort of up to the microbiologist, up to the scientist to try to determine and to try to pick some sort of parameters that we want to use to sort of justify honing in on, say, that specific organism of interest. So is it something like, the temperature of your incubator? Are you going to justify throttling that up or down based on maybe the internal body temperature of the animal? Like I've recently found out that camels, they run a little bit hotter. They're like 39 C. Or if we're working with reptiles, they're a little bit cooler, so we might throttle that down to 30. So, we might mess with temperature. We might mess with other things. It might be a substrate or a pH or some sort of osmotic pressure, an antibiotic. So what sort of things are you looking to include in your AGF enrichments? What helps you sort of get the actual AGF out of that fecal material when you drop it in a test tube with media in it?
So that's actually a question that could be its own episode, because we know so little about what anaerobic fungi really need to survive, like in detail. But we do know, like we have a couple of different media types that kind of work very well. And then we do know all of those mammalian AGF anaerobic fungi that came out of mammalian hosts seem to like 39 degrees fine. Like it's a pretty narrow temperature range between like 37 and 43 maybe, but 39 is often the best. And the pH, for example, is also pretty narrow. Like the pH they can survive in or thrive in is around like 6.5 to 7 is usually the max. I've tried lower than 6.5 and I've tried higher than 7 and it usually didn't work. So those two factors are kind of fixed. And then when we go for enrichments, we usually, I mean, the temperature, as you said, if we're enriching from reptiles, we go for 30 degrees instead of 39. But if we do mammals, we go for 39. We even tried for birds, and we can maybe talk about that later. They also have a higher body temperature. We tried higher, like 41, 43. But 39 is usually a safe bet other than the reptile strains. And then we usually keep the pH the same. The media composition, we could do it with or without rumen fluid, but the general media composition is pretty fixed. We only take away the rumen fluid or add it. And what we really vary mostly is the carbon source. So our fungi are cellulolytic, which means they degrade plant material, celluloses, hemicelluloses, and stuff like that. And depending on what transporters they have in their cell membrane and what enzymes they have, they can degrade different kinds of carbon sources, different kinds of sugars. Like wheat straw is a safe bet because it has everything, and it's like the natural carbon source that they usually come across in the gut of those animals. But some can degrade, I don't know, silane, while others can't, or some can degrade, like grow on galactose, while others cannot. So varying the carbon source is our safest bet at the moment to try to pull out different fungi out of the same sample. But yeah, so enrichment-wise, we usually vary the carbon source.
Okay, so
What about
carbon
you?
source. Well, before we jump to me, I've got a couple follow-up questions because I know the people are going to want to hear more, and I kind of do too. So you mentioned that carbon source is probably the most important variable that you all switch things in and out when you're trying to enrich for AGF. I've noticed that, kind of like you mentioned, things that perhaps these animals are eating. Oftentimes I see you put like a whole entire plant in the tube, right? Which is kind of strange. I'll sometimes do that, again, especially if I'm trying to look for a particular hypercellulitic type organism. But for the most part, like I want to stick something in there. I want it to dissolve nicely and, be clear and everything's hunky-dory. But for some reason, I see you guys literally putting like an entire thing of grass or an entire thing of straw or an entire thing of some other plant, like in these enrichments. And is it something special about just like having a whole leaf in there? Or, you know, what is it? What's the deal with that?
So actually, that's the main speciality of our fungi. Is they are anaerobic organisms that can really degrade, like attach to, mechanically break up, and enzymatically degrade plant material, like the whole thing. Because that's usually what anaerobic bacteria are not as good as. They usually degrade like the celluloses and hemicelluloses that are released when you break apart the plant material, but they're not really apt degraders of like the whole thing if you just give them the whole thing without breaking it apart.
Okay, so they really like to physically attach to that substrate that's in the tube.
They have like filaments or holdfasts, which are like little extensions with which they grab onto the straw or the grass or whatever. And for some reason, that's something I've noticed in because I experiment a lot with like different media types and whatnot in my culture collection to find like a sustainable, less expensive and less work intensive media to keep a culture collection alive. And for some reason, the moment you add straw to a media or like any other plant material like switchgrass, whatever, like the whole thing, usually milled, not really a whole leaf, but you mill it down usually. But still, the whole plant material, if you put that in, the moment you do that, those fungi grow more stable, I want to say, over time and can be sustained longer. So usually if I have plant material in there, I only need to subculture them once a week or twice a week, once every 2 weeks. So,
whoa, okay.
If there's like whole plant material in there, as opposed to if there's only cellobios, which is our go-to dissolvable carbon source, that usually requires twice a week instead of just once a week or once every 2 weeks subcultivation in like 10 milliliter tubes. If you have like bigger tubes, you would need to do it less. But yeah.
So is this a difference between getting like McDonald's versus going to a Michelin star restaurant and the 15-course meal taking you 6 hours? And is that what's going on here? Like I'm hearing you toss more straw in there and we're like, we're good for 2 weeks now.
I haven't thought about it that way, but that sounds pretty spot on, I think.
Okay.
At least in my mind, I have no proof or experiment or whatnot to really back that up. But I've noticed the stability of the cultures on straw, and I'm thinking like maybe the mixture of different carbon sources and the way they have to like express enzymes and whatnot to actually get to the good parts. Maybe all of that physiological activity is better for them than just tossing sugar at them.
Wow, look at that. Eat your veggies, kids. Eat your veggies, especially if you're an AGF.
Yes.
And also, if you're harboring a Neocalammastica mycota out there and you want to send it to Julia, by all means do. She's the queen of the culture collection. So she's looking at me like, I hate you. I have enough. They're all ours. But you do mention that, and we talked about that in an earlier episode, that like you maintain the largest collection of AGF on planet Earth. You know, perhaps there's one in a neighboring galaxy, but you know, as far as I'm aware, it's like the biggest one in the universe. So kudos.
Yeah. Okay.
World champs.
At least as far as we know. There are other labs that also work with anaerobic fungi, but I don't know if they have as many genera as we do.
Okay. Wow. Man, I don't even remember what you were going to throw the question back at me as. So I got really excited about substrates.
Well, substrates. So what is the substrates is our environmental cue that we vary the most usually. What is yours? When you enrich for bacteria, like what environmental cues are you looking at?
I think for me it depends entirely on what I want to try to pull out of this enrichment. Because oftentimes in my experience, I've found that bacteria really participate in a lot of like cross-feeding. And so say, for instance, I want to look for like xylan degradation, or I want to look at a particular, like if we're just talking substrate utilization and I switch out different substrates, a portion of the bacteria that are in that enrichment are going to utilize that sole substrate. They'll metabolize it, they'll spit something else out, and then the rest of the community blooms and flourishes on that secondary metabolite. And so while we do sometimes use substrates, it's probably not the biggest parameter that I find myself switching. I think for me, it's usually almost like a consortium of different things. So you mentioned that like with the AGF, there's a very tight pH band and a very tight temperature band. And I would imagine the same is probably true with salt content as well. I think with bacteria, it's a little bit more forgiving that they're able to grow. Like if something grows at 37, it's probably going to grow at 30. It might grow at 45. Like there's a pretty big band there. Same thing for pH. Like if it grows at 7, it's probably going to grow at 5, and it's probably going to grow at 8. And then salt, there's usually a couple % weight per volume that these things can vary. So they're pretty forgiving. But when I start manipulating multiple things at the same time, I think that tends to be when the magic starts to happen. But to give an example, like say of a recent enrichment, we've got a client at the MCCF, so that's the Microbiomics and Culturomics Core Facility here at Oklahoma State, for those of you that didn't already know that. Shout out MCCF. Go follow us on LinkedIn, Facebook, Instagram. One of our clients is very much interested in Bacteroides. And so Bacteroides is this genus of really, really, really cellulitic, obligately anaerobic bacteria that don't form endospores. So it's kind of problematic in that sense. But what I do for those enrichments is I start out with sort of a minimal media with something that is very, very difficult to degrade. Like I've literally put like Wattman filter paper in these enrichments for them to munch on, chem wipes, just some sort of not powdery cellulose type situation, crust up, crustacean, chitin type stuff, just to try to really let the microbes like work to degrade that substrate. And then oftentimes I will pair my enrichments in sort of 2 phases. So I'll have like an initial enrichment and then an enrichment after the initial enrichment. And so I'll take, an aliquot out of, say, my enrichment, put it into something else, and then try to re-enrich or subsequently further enrich for the specific thing that I'm after. And so I might go from like highly cellulitic to something, in the case of Bacteroides, that contains like, say, a lot of bile, because they can really tolerate a good deal of bile. And so in that way, I oftentimes find myself doing like multi-pronged enrichment approaches. But it really does tend to depend on the specific project in question. Like in your case with the AGF, it's like throw every antibiotic at it that's antibacterial because we hate bacteria. And like, we're super broad spectrum, all the vancomycin, all of this stuff, just juice it up. And then for me, I'm like, maybe I'll knock out the gram positives this time, or I'll, knock out some of the gram negatives, or I will try to, that sort of thing. So I find myself doing a lot of tweaking to various enrichments. Yeah.
Maybe for those of you who are not, or for those of our listeners who are not as deep into enrichment and isolation, we should maybe define what an enrichment is and what an isolate is. Because I've talked to people about enrichments and isolates, and for me, there is a clear difference. And I think for you too. But maybe if English is also not your native language, maybe it's not as clear to you. So what is an enrichment versus what is an isolate?
So I would say for me, an enrichment is a narrowing down or a funneling of a bunch of different microbes, a particular community, into something more selective. But it's not an individual unit. Like if, we're throwing out hypothetical numbers, let's say there's 500 different bacterial taxa in this sample that I'm working with, but I'm only interested in a fraction of that. I will try to enrich for the fraction, and after, say, the first enrichment, maybe I'm at 250. Then after the second enrichment, maybe I make it down to 100, but I'm still only interested in a fraction of those 100. When I have an actual isolate, for me, it's like an individual dot on a plate that is a, quote, well-isolated colony that I will then subsequently purify and identify and figure out, what is this thing? Is it the thing that I was after, yes or no? So yeah, for me, enrichment is sort of narrowing down, and isolation is when it's just one single unit. Like I've got one dot on one plate.
Yeah, absolutely. So enrichment is usually the first step to isolation, but an isolate is the end goal, while the enrichment is just like, as you said, like narrowing it down. So when I start an isolation process for anaerobic fungi, I take 4 tubes with media. I put my fecal sample into the first tube, and then I dilute that first tube serially into the other tubes. And those are my enrichments. And from those, I then, when I transfer and do roll tubes and whatnot, that's how I gain an isolate. So what does that process look for you?
Yeah, that's a fantastic question, because we do this so differently. So usually I oftentimes also will start with a dilution of some sort. What I do is I usually get the sample in, and then I will do, say if it's a fecal sample, just for simplicity's sake, I'll do about 1 gram of the fecal sample in about 10 mil of, like 1 gram and 9 mil of anoxic sterile PBS. And then I'll just kind of give it the old shaky shake, vortex around in the chamber. And then from there, I will go into an enrichment directly. And I typically go into liquid enrichments. So I don't oftentimes enrich on plates first. And so I will let the bacteria cook in that enrichment at a plethora of different parameters. So I may have probably somewhere in the neighborhood of like 60 different permutations of enrichments going on simultaneously for like one sample. Because when you start thinking about manipulating salt, pH, all at different temperatures, all with different substrates, it gets ridiculous very quickly.
Yes.
The serum bottles and the Boltz tubes get out of control. But I go into the liquid for the enrichment, and then I will let it incubate for some period of time. Usually it's anywhere from like 5 days to 2 weeks. But oftentimes what I will do is over the course of that enrichment, like say every day, I will take a sample out, and then I will do something with it, whether it's taking that sample out and putting it in another enrichment, or taking that sample out and just plating it to see what it might look like with respect to like individual isolated colonies. But that's typically what I'm doing with an enrichment after I dilute the material in PBS.
Interesting.
Hence my love for phosphate buffered saline.
Thank you for explaining your process to us, because that was very interesting for me to hear. And it kind of brings me to the next question you jotted down in our script. Because the question says, why is isolating AGF uniquely chaotic compared to isolating anaerobic bacteria? But listening to you and your 60 different enrichments, I feel like I have a pretty cool, chill life here. No chaos. Because usually my enrichments are 4 or 5 different things, not 60 at once. So,
well, I'll just keep tooting my own horn here. So you imagine those 60 enrichments, right? They ultimately need to get plated on something. And the psychopaths in the anaerobic bacteriology world don't just plate them on one media. So you're thinking you've got 60 enrichments, time points of these 60 enrichments. Each time point is going on 5 different media.
Jesus.
Potentially. Not that that's the case every time, because again, there's different levels of psychopathy, but sometimes when it necessitates, it can get wild.
Jesus. I mean, I would love to test more, but it's just there's more work involved. Because not that I say bacterial work is not work, that sounds crazy to me what you're describing. But whipping up a set of plates takes an hour in total. You first put everything into a flask, put it in the autoclave, and then the pouring is the longest step of it, and that takes max an hour maybe.
Yeah.
And if I think about anaerobic fungi media, where it takes up to 2 hours to actually prepare it, put it into the autoclave, and then sometimes when you take it out of the autoclave, you then have to add more stuff. If it's minimal media, you have to add the vitamins afterwards. You definitely have to add the antibiotics. So the whole process of media preparation without the autoclaving step would be 3 hours per 1.5 liters.
Yeah, no, you're exactly right. And even sometimes, and that's why I try to tend to avoid overtly complicated media. I will make it when when it's a necessity. But, if I don't have to add a filter sterilized anoxic vitamin stock solution to something and a this and a that and the other thing, and I try not to. But sometimes I have to. I have to do that. And that, as my old boss would say, that drives me crackers. So, shout out Paul Lawson. If you're listening to this, buddy, I love you. Thank you for putting up with me for 6 years. But yeah, so I think, when I wrote that question down, I was really thinking about how oftentimes as you're trying to isolate things, you inevitably isolate bacteria instead of fungi.
Yes.
And I think part of that is just the numbers game, because there's probably in the population, it's probably orders of magnitude more bacteria than fungi, than AGF.
Yes.
And so I think that, to me, makes your process so much more complex, and especially from a contamination standpoint. So say as you're working with the sample, you're not really going to contaminate from your hands with AGF.
Usually
I wouldn't
not,
think, you probably don't have,
yeast is your biggest enemy, I think. And those usually die after a while. They don't really
they wouldn't be obligate anaerobes. But with the bacteria, it's if I don't know, look at the Eppendorf tube wrong in the chamber, I've contaminated the Eppendorf tube with whatever was on the glove, because the bacteria be everywhere. And so, I think for me, the AGF is just all mitigating chaos. Maybe that's just enrichments in general. It's all just chaos mitigation.
I don't know if that's not just life in general.
Yeah, I don't
mitigating chaos.
Yeah.
Going back to your point, I think for AGF, it's a numbers game. So the bacteria are really our biggest enemy. And I don't know what's happening, but the last couple of weeks, I've been going through a dry spell. And even from horses and cows and deer, which are canonical hosts for AGF, where it's usually very easy for me to isolate AGF from, I'm not getting anything. I'm only getting those stupid bacteria. And they masquerade as fungi sometimes. That's also because there are certain growth signs that you look out for the fungi display.
Yeah.
Like if you have plant material in there, the straw usually clumps together when fungi are growing because they like all attach to the straw and to each other, and then they form like big mats. And gas production and biomass production and whatnot, and a specific smell once you punch the needle into the bulge tube. But there are freaking bacteria who would also clump the straw and make it float. And I've isolated those now, I don't know, 5 times. Of course, I don't know if it's an isolate. It's an enrichment probably. It's probably many different ones. And I don't know if it's even the same ones. I have not dumped them on you yet. Usually that's what I do when I get a bacterial isolate. I'm going like, Sam, are you interested in this bacteria?
Yeah, and I will say, in a way, that's probably a giant portion of what I typically do in the lab is just kind of piggyback off of, dare I say, like your failed enrichments. And failed's the wrong word, but it's like the enrichments.
It's the right word.
It's the enrichments that turned up more bacteria than they should have. Like, again, you're hoping for the AGF and you get something else. And then it's like, this dude, he's weird. He's interested in these bacteria. And then I'll go and take a peek at them and see sort of what's going on. And sometimes, depending on the animal, depending on the enrichment, how many times it's been passaged, et cetera, sometimes I find just sort of like one colony morphology. I'll just directly put it on a plate. And it's like, whoa, it looks like there's just one thing in there. Or I'll get it and I'll plate it and it'll just look like, Coachella. It's just ridiculous. And I'm like, woo, what is happening here? I need to dilute you. So break out the PBS. But yeah, so I do enjoy when I get those little, tubes of those enrichments that sort of went not the AGF way. And we also, have a manuscript that was recently submitted that is essentially detailing a novel bacterial isolate pulled out of one of these AGF enrichments. So yeah, we're getting some actual stuff out of it, which is pretty cool.
Yeah.
So I think.
Yeah, and I think it's unique in this kind of, like, our lab in this way is very unique because I don't think there is, or at least I'm not aware of another lab like that divided between the kingdoms and has like fungi and bacterial world combined. Because usually it's either like when they're working on anaerobic fungi, the other part is usually archaea, because anaerobic fungi and archaea usually form a kind of syntrophy, and then usually that's the collaboration you have. But we're trying to isolate fungi, and you are focused on bacteria completely outside of the fungal realm. Like, we don't care if those are syntrophic to anaerobic fungi or not. Usually they're not. And then, yeah, I think that makes our lab kind of unique.
Definitely. I totally do agree. And my only regret is that I've yet to give you an AGF. I'm like, would one of those ever pop up in one of my enrichments? And then I go, obviously not, because of all the things that you've been talking about, the necessity of a certain temperature and a certain this and a certain that. And I'm always messing with everything. So I'm like, there's no, I've lost them before I even started.
But you've pulled out some fungi yet. Maybe we just need to expand our fungal kingdoms that we are, like our fungal phylums that we are working on.
Yeah, there we go. I'm down with that. Because we do isolate those every once in a while. But typically, they're aerobes. So.
Yeah.
Less cool. Oh, man.
Well,
where does that put us?
I think that puts us right into the path to our game.
Ooh,
if
game.
you want to.
I'm sure Julia will come up with a better sound effect than that. So.
Oh, no, that's our game sound effect now.
Ooh, boy.
So, people, today we've brought, in addition to our lab yaps and the questions from the undergrad, we have another game to play. So, the game is called This or That, and the rules are simple.
That's good.
I've brought a list of this or that questions. Like, it's 2 things, and you are going to pick one of them.
Okay.
You're not allowed to ask any follow-up questions.
Ooh.
And you're not allowed to give explanation on why you picked one over the other. We're just going to rattle through them quite quickly.
Quite quickly.
That's
not one of my strong suits.
And no explanation needed on why you picked one over the other.
I have a follow-up question. Am I
not
allowed to
allowed
say
to.
both? Or am I allowed to say neither?
No.
Or do I have to pick one?
You have to pick one.
Okay.
Your life depends on it.
My life depends. Well, it's been a good run.
Okay, Sam.
All right.
I've actually brought
ready.
this. Yes, I actually brought this on paper,
Oh my.
so you cannot peek at it. On
we are old.
Yes. Okay, so first we're going to do a couple of warm-up ones.
Okay,
so they're about the most important thing in the world, which is snacks.
Ooh.
Are you ready?
I'm ready.
Okay. First one, salty or sweet?
Salty.
Salty or sour?
Sour.
Soft candy or hard candy?
Soft.
Dark chocolate or white chocolate?
Dark.
French fries or mashed potatoes?
Oh, cursed. French fries.
Nice. Okay, and then let's get into the more relevant ones.
Oh, those weren't relevant?
Oh, I mean, they are relevant to find out who you really are, but now we're going to get into the weeds.
Ooh, okay. Into the weeds we go.
Into the weeds we go.
Come with us, kids.
DNA or RNA?
DNA.
Illusion buffer or water?
Elution buffer.
100 microliter pipette or 200 microliters?
200 microliters.
What? Okay. 10 or 20 microliter pipette?
10.
Single or multi-channel?
Multi-channel.
Short tips or long ones?
Long ones.
Benchtop versus anaerobic chamber?
Chamber.
Anaerobic chamber versus hypoxic chamber?
Ooh. Dun, dun, dun. We have both at the MCCF, folks. Anaerobic chamber.
Anaerobic chamber versus podcast chamber?
Podcast chamber.
Lab work versus data analysis?
Lab work.
Lab work or paper writing?
Lab work.
Lab work or reviewing a paper?
Lab work.
Reviewer 2 or the PhD committee?
Oh. Reviewer 2.
Okay. Plate or liquid culture?
Plate.
New isolations or characterizing a novel isolate? Like setting up new ones or characterizing one?
Oh, my. Ooh, this is tough. I'm picking between my children here. Setting up a new enrichment.
Nice. Yeast extract or beef extract?
Ooh, beef.
Beef. Or
That's what's
blood.
for dinner.
Beef extract or blood agar plates?
Blood auger plates.
Blood agar plates or McConkey agar?
Blood auger plates.
Aerobic or anaerobic culture?
Throw this question out. What are we even doing here? Anaerobic. Come on.
N2 or CO2?
Into.
N2 or H2?
Into.
Okay, so the last three,
Oh, no.
E. coli versus Bacillus subtilis.
E. coli.
E. coli or Saccharomyces cerevisiae.
Get out of here with these yeast. E. coli.
Archaea or fungi.
You said neither wasn't an option, so I'm going with fungi.
Wow. Okay, last one. Fungi or bacteria.
Bacteria. So I'm going to attempt to throw this back to you, and it will be a lot less insightful than what was just thrown at me. So,
Oh, you think we've figured out who you are deep down?
no, that's locked away somewhere. Okay. So, this or that with Julia. This is the judgment zone, folks. Here we go. Birds or reptiles?
Birds.
Birds or mammals?
Birds.
Birds or fish?
Birds.
Birds it is. Coffee or
Coffee.
soda?
Coffee.
Oh, okay. Coffee or tea?
Coffee.
Coffee or energy drinks?
Coffee.
All right.
Wow.
We're going to caffeinate these birds. Okay. Playing sports or going to an art gallery?
Playing sports.
Playing sports or going to a museum?
Playing sports.
Ooh. What sport? Wait, we're not doing that right now. We'll circle back to this. I'm bad at this game. Clean or clean-ish?
I think everybody who knows me knows the immediate answer to this one. Clean.
Online shopping, in-store shopping.
Online shopping. I hate people. Oh, sorry. No explanation allowed.
But not the listeners of this program. We love you all, and we appreciate all 7 of you. Thanks, Mom.
Thanks, Mom.
Microwave dinner, fast food.
Microwave dinner.
Microwave dinner or fancy restaurant.
Fancy restaurant.
Fancy restaurant or conference snacks.
Fancy restaurant.
Depends on who's paying.
Yes.
Let's see. This or that. Go to bed early or finish and submit manuscript.
Oof. Jesus. Finish and submit.
Reviewer number 2 or broken -80.
I like how we both included that one. Reviewer number 2.
Cold room, hot plate.
Cold room.
Clean desk, controlled chaos.
Controlled chaos.
Fresh gloves, unopened pipette tip box.
Ooh, unopened pipette tip box.
I'm inclined to agree with that one, even though I can't say that. And the last one that I have, roll tubes or liquid cultures.
Liquid culture.
Okay. Okay, we'll have to elaborate on some of these answers at some point in time. I did try to copy you a little bit. I'm like, I got to throw these back.
I think you did
especially
a good job.
the food one, it was basically like microwave dinner or conference snacks. Like that's where that one started. And I went, well, I got to say something else. Can't be that guy.
You made them up on the fly now?
Probably about a third of those.
Wow.
Yeah.
Look at you.
I'm
I wouldn't
a man
have
of
guessed.
many talents.
Yeah.
The English saying there would be the jack of all trades, master of none.
But better than the master of some.
That's right. Most people forget that last part.
Yes.
Oh, my.
Well,
that was good.
fun.
Yeah, that was good.
I like that.
Yeah.
I hope our listeners like that too.
Well, if we haven't, brushed them away yet, we've, another opportunity arises.
Yeah. Everybody who stuck through the smell episode will
Thank you for that. We owe you a Christmas card.
Yes.
Oh, my.
Oh,
Where does that
wow.
leave us off?
Let's see. Oh, man. We actually touched on most of those.
Look at us go.
Well, let's say we've got our enrichment. We're trying to get something new. At what point would you know that it's time to stop the enrichment or the enrichment trials for your bacteria and start to really isolate one single colony?
I'm going to get real with you for a second. I'm going to get real close to the microphone here. I have a sickness. And the only cure for that sickness is more. And so I am not usually ever inclined to stop trying. That being said, I think at some point you have to draw a proverbial line in the sand. And you have to go and have a little mental pep talk with yourself with the devil on one shoulder and the angel on the other. And it's like, Sam, buddy, we got that new phylum of something out there. And then the angel's like, but Sam, I don't know what the angel was going to say. It's like, dude,
It never gets to talk.
you got 40 enrichments going already. Do you know how many plates that is? You can't even do 40 times 4 in your head. No, I think at some point you have to have some guidance. Typically what I like to do, because I know this about myself, is I draw up sort of an enrichment strategy before I start, and then I try my best to stick to that. I will go, I'm going to mess with these things, and I'm going to mess with them in this way. And I might ad-lib a couple on top of that in the moment if the mood strikes me. But for the most part, I try to stick to that plan. Otherwise, it just becomes chaos. more chaos than it already will be.
Controlled chaos.
Exactly. Well, it's not even controlled at that point. It's like off the rails, anarchy, burning down buildings. It's rough. But so I think, yeah, for me, enrichments, they sort of stop when my imagination stops, and I just have to force myself to be unimaginative at times. But as far as when it's time to start doing subsequent investigations, I like the back end of that question, because it's like, ultimately, what is the point of enriching for and then uniquely isolating something, as an individual unit? Why? What's the point? Mm-hmm. Because I think sometimes I'm met with criticism about, like, oh, yeah, Sam, cool, but there's bajillions of things in this environment, in this niche. Why does one of them matter? And what I was told and what I believe is that, yeah, we can do like metagenomics and we can look at all the data that are out there, and we know that they're complex communities and all of those sort of things. And we can make predictions, right? But I don't think it's the end-all be-all, in a sense that genotype is important, but it doesn't always reflect phenotype. Mm-hmm. And then phenotype is sort of something that is about when is it expressed in a given environment. And so without isolating something as an individual unit, you can't really probe in and test those questions, mm-hmm. or even put it in a biculture, a triculture, a polyculture of some sort, and then attempt to ask questions like that. First, you have to isolate the thing. You have to figure out the parameters, what can it do, what can't it do, and then force it to do things, challenge it with certain factors, and then, again, as you're describing what that thing can and can't do. Mm-hmm. So I think for me, that's how I would long-windedly answer that question. The this or that's got me wanting to talk.
Yeah. And anaerobic fungi isolation is, while it's more complex in some ways, it's so much easier in other ways than bacterial isolation. Because, in anaerobic fungi world, the isolation process or the enrichment process stops where the bacteria take over, which is usually fairly quickly, because bacteria have like a generation time way faster than our anaerobic fungi. So you usually know very quickly if you have to stop an enrichment or not. Because once the bacteria take over, usually our fungi can't do anything about it. I usually, sometimes when it's like an important sample, or when I have a lot of hope, or I don't know, when it's one of those days where I can't take another loss, I will go in with another set of antibiotics and try to subculture, try to rescue our fungi in some way or the other. But usually once the bacteria take over, this enrichment is done. You have to start over. Mm. While our fungi are more complex and more fastidious and harder to keep alive and take longer to grow, so every isolation attempt you do, it takes you at least a week, if not 2, until you know whether you found something or not. But yeah, in this way, it's easier because it just stops when the bacteria take over. When you see turbidity and smell those grimy little things, it's done.
Yeah, that's so interesting that the turbidity for you guys in the lab is sort of a no bueno. That's something that you all are not really interested in. Whereas I walk in and I see a turbid tube and I go, yes, success, eureka. So that's I think that's interesting.
Yeah, absolutely. And it kind of healed because every time I see the bacteria now, I'm thinking like, maybe this could be something interesting for Sam, instead of thinking like, oh man, another failed experiment.
Aw. Well, thank you. I think we got when to stop enriching and when to actually isolate something. But how do we actually know that we've only isolated one thing?
Oh, yeah. That's a good question.
You think that you've got it. You think that it's all in there all alone by itself. How do we verify that there's only that one thing now in that tube on that plate? How do we do that?
How do we know?
How do you do that? Because I know how I do that. I tend to run some basic things, like I'll just do like a wet mount, little phase contrast, take a look at the cells, and I'm like, oh, do they all look the same? Yes or no? If I see a bunch,
is that enough in bacteria? Because I feel like
not always
it's like
enough, but it's like a good, like if I do that and I see like there's circles and then there's rods and then there's different sizes of rods, I go, oh no, I have really messed up. But if I, flip on the phase contrast and I just see like a bunch of circles, and they're all conglomerated together in tetrads and diploids or whatever it is. And I'm like, oh, like that's a good indication. So I'll usually start off with some just basic good old phase contrast, and then I will move into doing like a proper gram stain reaction. The colony on the plate or from the tube or whatever it might be, and then just, again, take a look at it. Like, do I see only purple circles? Do I see only pink rods or purple rods? Or, again, are they all the same size? Do they have the same sort of cell orientation? And like, that's probably the biggest one for me. On top of that, what I will then do is I will run and sequence a particular gene from my what I perceive to be as well-isolated bacterial cultures. And so for me, and for all of those bacteria folks out there that are hanging on, will know well, very much so, that it's the 16S rRNA gene. And so it's about 1,500 base pairs, and I'll get that little sucker Sanger sequenced, and then I'll take a peek at it. And if I've got nice, clean Sanger reads with nice tall peaks and not a lot of mess, and everything's pretty high quality and things are looking good, then I will know, hey, this probably is pure culture. And then eventually it gets, whole genome sequenced and all that jazz where any nuances are teased out. But yeah, usually for me, it's a little bit of microscopy. Well, actually, I guess it starts with a plate. It's like, does everything look uniform on this plate? Same size, same color, same margin, same elevation, all of those morphotypes. Then it's microscopy, 2 different ways. Then it's 16S sequencing.
Nice.
Yeah. What about you?
Would you be very much surprised if I say it's pretty much the same process?
What?
Only that, of course, it doesn't start with a plate for us, because, come on. But it starts with our version of a plate, which is a roll tube, essentially just a tube that has the agar slapped onto the walls. So you can also grow colonies on it. And I also check, like, how does the colony morphology look like? Are they all the same type? Do they have the same color? Are they fuzzy or not? And whatnot, because some fungi are, some aren't. And then microscopy. Usually I do microscopy on the roll tube, which is pretty cool. You can like take a closer look at the colony with just a normal microscope.
Okay.
And then you already see, like, are all their little sporangia stuck together and their filaments outwards, or are there like bulbous ones versus filamentous ones and stuff like that? You can very easily detect with microscopy, either on the roll tube or just on liquid culture. Then you can do some DAPI staining. So stain the nuclei with a fluorescent dye that shows you where the nucleus is located in the fungi, and that sometimes helps you to determine whether there's different things in there or not, because some fungi have them in the filaments and the sporangia, and some have them only in the sporangia. So colony morphology, microscopy. I also look at the biomass formation in liquid culture, because those also vary between the fungi. And usually after 1 or 2 rounds of colony picking, if everything looks uniform, if everything looks good, I do Sanger sequencing as well. We don't go for the 16S RNA, because of course our fungi don't have them.
Sacrilege.
But the gold standard for anaerobic gut fungi is the D1, D2 region of the large ribosomal subunit.
I will take your word for it.
Which is a 700 base pair piece. And we Sanger sequence this one. And then, as you said, if everything looks good, high quality, high peaks, no noise in between, we're happy. And then that's where the differences come in, because genomics, not going to work. Or it is, we'll see. We're working on it.
Yeah.
But yeah.
And that's something that we should talk about at some point in time is why it's so inherently difficult to sequence the AGF.
Oh, yeah.
That might be a good topic to bring up with a future guest on the podcast. Wink, wink. Stay tuned for more of that, kids.
Cliffhanger.
Yeah, cliffhanger. Tune in. I'd say next week, but just keep tuning in. Eventually it'll happen.
Yes.
We'll make it make sense. Yeah, so that's interesting. So it's basically like, what's it look like in the on the agar, microscopy, and then a little bit of sequencing.
So the general outline is the same.
Yeah. Well, look at that. We're not so different, but we are. Interesting. Wow.
Yeah. Well, that's the word of the day. Or as we would say in German, that's the word of the Sunday.
The word of the Sunday.
It's like, because the priest would like end his...
I see.
His sermon is like the word of the Sunday. So when you say something very profound or something that would like...
Okay.
At that point, it's like, all has been said. It's like the word of the Sunday.
All has been said. Man, I am always impressed by how long we talk about things.
Oh, yeah.
And we should let our audience know, Julia, that as of yesterday, so not that anybody knows when that is, but as of mid-November, I have no idea when this is going to be coming out.
I thought we keep it a secret and we just keep it as of yesterday.
Oh, okay, there we go.
The magic yesterday is whenever they listen to the episode.
Perfect. So Julia has cut that last part out. As of yesterday, we actually have now an internet presence. So we are alive and well on the web. You can find us on several different social media platforms like Blue Sky, Instagram, LinkedIn, YouTube. I think that's it, as Life Unculturable. And then we also set up an email. So we would love to have, not that we hate our questions from our undergrads, but, we probably should expand the sample size just a smidge.
So if you have undergrads too, and they have questions.
Yeah, they want to know about the birds and the bees of anaerobic microbiologies, send them our way at lifeunculturable@gmail.com.
Wow.
So, yeah, that's out there and about there.
And maybe also the thing that sparked our internet presence is actually Michael and his work.
Oh, true. Yeah, I meant to give him some props. But I should probably let you do that because I don't even know what Michael's last name is. And if I did, I couldn't pronounce it. Sorry, Michael.
So actually, in addition to Sam and me, the voices you hear every time you tune into the podcast, we have another member of the team, Michael, or let's say the name in German as well. So it's Michael Prattes, Dr. Michael Prattes. Michael from Austria, who is he's also a microbiologist, also a postdoc, but he fled the scientific world, the research world itself, and he became a graphic designer and is now working in science communication, which I think is awesome.
True.
And he's a
really
brilliant, he's a brilliant and creative head, and he actually designed our logo that we have in the Life Unculturable podcast. And that, the finishing of that logo is what sparked our internet presence.
Entirely in and of itself. Yeah, we were out in the middle of the ocean in a dinghy prior to having that logo. So if you love it, you should show them some love. Yes. And if you hate it, you should just pretend that I did it. So that works for me. That works great. That's usually what I tell people when we go to a cookout. They'll be like, Sam, who made this? Did you make it or did your wife make it? I'll be like, well, if you like it, she made it. If you don't, I made it. We'll just leave it there. Oh, my. But yeah, serious thank you to Michael. And serious thank you to you all for listening to us and hanging in there with us. And then we look forward to interacting with you online now. Yeah. You can hopefully expect at least periodical updates from me or Julia on the socials. That seems weird to say. I'm not cool enough to say, check us out on socials. What?
We already established that it's internet presence, not social media.
Yeah. Check us out on the internet, kids.
That sounds about right.
Yeah, buddy. Oh, my. Well. And then did you want to do that outro? I saw that you had queued up
I did.
An outro down at the bottom.
I did. Oh, I did.
Yeah, look at that. Come on now.
It's been late yesterday.
Two options. Well, see, that's why you were finishing things up instead of going to bed early. I know this about you now. Let's see.
Well, Sam, after all that yapping about the enrichments and the isolations and all, I'm kind of itching to get back to the lab and see what other novel organisms we can magic out of our freezers. What about you?
I don't think I could have said that any better, Julia. I say we get back to the lab, we check on what's cooking. Yeah. Thanks for hanging in there with us, folks.
Thanks.
We look forward to jumping into your ears in the very near future.
Yes. Yeah. Thank you for spending time with us, and see you all soon.
Yeah.
Or hear you all soon.
Ooh, there we go. Bye, everybody.
Bye. Also, we're getting back to your not favorite chamber, because we now know that the podcast chamber beats the anaerobic chamber.
Well, I mean, it's less weird to talk in the podcast chamber than it is when you're just in the anaerobic chamber and nobody else is in the room with you. And you're in there mumbling to yourself.
Mumbling to your microbes.
Oh, yeah. I'm micro mumbling all the time. Come on, guys. I'm never